from Osaka Medical and Pharmaceutical University, and she will talk about membrane potential modulates ERK activity and cell proliferation.
So first of all, I direct to thank you for the inviting me and giving me an opportunity to talk my recent work. So take-home message of my talk today is the membrane potential is not only for ion channel or not only for neuron. Membrane potential work diverse function as we thought. So today I will talk about some data showing membrane potential modulate ERK activity and cell proliferation. So what is the physiological function of the membrane potential at non-excitable cell? Is it only for ion channel or only for neuron? So hypothesis is that the membrane potential may have physiological function other than action potential. So there is some example indicating that membrane potential work as other than action potential. For example, paramecium, this is small living thing. It's very famous in Japan. It's called zorimushi in Japanese. And in Japanese kids know about this small thing because they run at school. This small living thing use membrane potential to move cilia. It's too small, but they're cilia. And they use this cilia to move. And when they want to change direction, they change membrane potential. Plants also use membrane potential. This plant, Mimosa pudica, it's called ojigiso in Japanese. The meaning is bowing plant. In English, I think it's called a silent plant. The leaf is closed when touched. Those movement is also regulated by membrane potential. There are some other example. Arabidopsis, they also use membrane potential. When caterpillar eat the leaf, that information is transferred to neighboring leaf. Here, there's electrode between E2 and E3. When caterpillar just walking, there's no membrane potential change. But when caterpillar eat leaf like this, then the membrane potential is changing, and this information is transferred to neighboring leaf as an emergency signal. And interestingly, this membrane potential change is converted into the response at the molecular level, such as stress hormone release. So the hypothesis is that could membrane potential regulate signal transduction such as ERK or AKT? And could membrane potential regulate cell proliferation, differentiation, or migration as a consequence? That is a question. Is there any linkage between membrane potential and intercellular signaling, something like this? So So there was some literature about relationship between membrane potential and cell proliferation. This paper published in very old 1978. In this paper, those cells with high mitotic activity have this. Sorry, this is very small, but those cells with high mitotic activity have depolarized resting membrane potential. And those cells with low mitotic activity, such as contact inhibited cell, have hyperpolarized membrane potential. There have been many reports about the relationship between membrane potential and cell proliferation. Mitotic active cells such as cancer cell or early embryonic cell have more depolarized membrane potential. And those cells have low mitotic activity, such as neuron glia or skeletal muscle. Those cells have more hyperpolarized membrane potential. However, there's no direct evidence that depolarization really induces proliferation or not. So we started to study if membrane potential really affects the cell proliferation. So to direct study the membrane potential, the effect of membrane potential on the mitosis, cells are synchronized. When cells are treated with thymidine, cell cannot enter to S phase, so cell can synchronize at G1S border. So experimental setup is like this. The cell were treated with thymidine, and after 8 to 9 hours releasing from thymidine, cells were imaged with confocal microscopy with thick mode. So mitotic cell is easily recognized like this. So mitotic cell count is accumulated for every 1.5 hours and plotted against the time after imaging start. Then 6 hours after imaging start, cell is going into the mitotic phase. When cells are depolarized with 15 millimolar high potassium solution, this is this yellow-brown one, mitotic activity is upregulated. Here, right graph is mitotic cell number is accumulated within 6 hours, so altogether in this 6 hours. The result indicates the same point that with 15 millimolar potassium situation, mitotic activity is upregulated. This effect is almost same level as cell treated with FPS. In this graph, pink and blue one. And this graph, this two one is FPS. And the arc is the key molecule to regulate proliferation. So next we investigate the potential involvement of arc on this depolarization-induced mitotic activation. U0126 is a MEK inhibitor. So when cells are treated with MEK inhibitor, in this graph, it's green and green-blue one. The depolarization-induced mitotic activation is abolished. This graph also indicates the same point. So next, we next investigate the relationship between the membrane potential and arc activity. So how to measure arc activity? So we use arc biosensor called ECA-EV. This sensor is FRET-based biosensor. When arc is active, FRET occur. And how to change membrane potential? We perfuse high K solution. This is an example. Please look at cell number 5 and 6. At the time point 10 minutes, we perfuse high K solution. And at time point 30 minutes, EGF is add. I repeat again. When high K is perfused, arc is activated, and with EGF addition, arc is further activated. So this is the result from several experiments. With high K perfusion, arc is activated. When I perfuse with 30 millimolar, sorry, I don't mention. In previous slide, it is 145 millimolar potassium. It's far from physiological condition. When cell perfused with 30 millimolar potassium, the arc activation is a bit mild. And with 50 millimolar potassium, it's much more mild. The purpose of adding EGF at the end of experiment is to measure maximum arc output for each cell. The arc activity with high K perfusion is B, is normalized with maximum arc output A. So B divided A we call percent arc activity, and they plot against the logarithm of the potassium concentration. So as you know, according to Nernst equation, membrane potential is dependent on the logarithm of the potassium concentration. Here, the correlation coefficient is 0.54, so which indicate that arc activity is dependent on membrane potential. Next, to exclude the influence of perfusion itself, and also to control the membrane potential accurately, the membrane potential was controlled by perforated patch clamp using this solution. In this experiment, HEC cell is transfected with ECA-EV arc biosensor and voltage clamped. Cell is holding at minus 80 millivolts for 5 minutes, then depolarized to 0 millivolts for 5 minutes. This red line is patch clamp cell, arc activity with patch clamp cell, and blue one is control from the same field of image, but not clamped cell. So with depolarization to 0 millivolt, arc is activated. But when membrane potential is returned to minus 80, arc is returned to the level that is in the beginning. Here is the graph. When cell is depolarized, here there's a significant difference of arc activity. And when it back to resting potential, here it's a significantly difference. So it means the arc activity is returned to the resting level. Yes. When cell is depolarized to -20 instead of 0 millivolt, the still arc is activated. However, when cell are at -40 millivolt, it's no more activation. It is because for hex cell, resting potential is a little bit depolarized situation, around -40. So for hex cell, this -40 millivolt is not depolarization stimulus. So this is summary. When cell is depolarized, arc is activated in the voltage-dependent manner. And when cell is returned to holding potential to -80, arc activity is decreased. So again, I plot percent activity against the membrane potential, and the correlation coefficient is 0.54, indicating that arc activity has membrane potential dependency. So when membrane is depolarized, in general, voltage-gated calcium channels open and intercellular calcium increase. Then this influence cause many diversity of the phenomenon. So next we check if that calcium influx is involved in this depolarization-induced arc activation. In this experiment, we perfused with 145 millimolar potassium, but calcium-free condition. The cell has still arc activation with high K perfusion, which means calcium influx is not involved in this phenomenon. Furthermore, there's some literature indicate that sodium influx is involved in cell proliferation. So we did the same experiment, but without sodium in extracellular solution. So we exchange sodium with NMDG. And the result is the same, that with high K perfusion, arc is activated. So what is the mechanism? We confirm the arc activation by Western blot here. And MEC and CRAF is upstream of arc. And all these molecules is upregulated by high K. And the RAS is the upstream of CRAF. So we next study membrane potential, a relationship between membrane potential and RAS activity. So how to measure RAS activity? We use a RAS biosensor called RAS. It's also FRET-based probe, but this time the FRET we measure by lifetime measurement called film FRET. The RAS activity measured by film FRET, and the result is here. The lifetime of CFP is decreased upon high K perfusion, indicating that RAS is activated by membrane depolarization. And this is a paper published in Science 2015. According to this paper, depolarization-induced phosphatidylserine clustering. This is picture taken by electron microscope. And according to this paper, RAS is interact with phosphatidylserine, and RAS also cluster upon depolarization. So we tested this hypothesis if phosphatidylserine is clustered in our experimental condition. So how to measure phosphatidylserine? We use a phosphatidylserine specific probe called LACT-C2 fused with CFP or YFP. And using the intermolecular FRET, when LACT-C2 YFP and CFP is co-expressed at same cell, the lifetime of CFP is decreased, indicating intermolecular FRET occur. So then we tested if phosphatidylserine clusters occur in the high K situation. When high K is perfused, CFP lifetime is decreased. That means depolarization-induced nanoclustering of phosphatidylserine. So, when we next test if phosphatidylserine depolarization affect this depolarization-induced arc activation. So when phosphatidylserine is depleted with phendyline, which deplete phosphatidylserine from cell, the high K-induced arc activation is abolished. So phosphatidylserine clustering is crucial for depolarization-induced arc activation. So this is summary, and we thank Dr. Matsuda for providing us arc biosensor and RAS biosensor. So thank you for your attention.
Thank you for this very stimulating talk. Any questions?
Thank you for extremely high impact presentation. And at the beginning, I thought maybe the effect of depolarization is simply mediated by calcium influx, but that's not the case. And it is truly exciting to show the clustering of phosphatidylserine, which is voltage-dependent, induces this event. And my question is, if clustering of phosphatidylserine could occur by depolarization, so it could influence not only RAS activation, but also other events. So do you have any evidence or speculation?
Right now, I don't have data, but I'm thinking the same. So next, my work is finding some other molecule that is affected by membrane depolarization. For example, SARC is also interact with phosphatidylserine. So maybe SARC is one possibility.
Maybe a question from the students or postdocs. At FASTEP conferences, we usually offer drink tickets, but we don't have them. So, but maybe somebody wants to volunteer. Somebody think about it. While in the meantime, we let somebody else ask a question.
Excuse me, thanks very much. I was just wondering, you've shown this in sort of epithelial cells. Do you think this process, the membrane potential regulation of phosphatidylserine, etc., what do you think the implications are for excitable cells like neurons or cardiac myocytes with the potentials going up and down?
In neuron, it's also known that ARC is activated by action potential depolarization. But in case of neuron, the calcium influx is involved. So I think it's a bit different phenomenon.
Trainees, students, postdocs. somebody else.
Thank you very much for a wonderful talk. So you said that the normal lasting membrane potential of 290 cells around minus 40. But in your particular experiment, you held the cell at minus 80. So have you done the experiment with the initial holding potential a little bit vaporized condition, minus 40 or minus 50 or minus 60?
No, when I doing patch clamp, I didn't think about that hex cell have vaporized resting membrane potential. So I keep like normal minus 80 millivolt, but I don't try minus 80. And
I hope you try it in the future.
Thank you.
Maybe I can ask a question. So you showed in the beginning that if you block ERK activation with an inhibitor, you can basically prevent proliferation, right? So there seems to be some specificity for ERK. But if you did, for instance, phosphoproteomics or some other unbiased method, how widespread do you think this effect of depolarization on signaling would be that is independent of calcium or sodium, right? If you designed an experiment where you either used calcium-free buffer and then did some phosphoproteomics, what do you think you would find?
You mean, is there any other than
pathways? Yeah, exactly.
Other than ARC?
Yeah.
I don't have, so far I don't have any data, and there's a possibility, but I cannot answer the direct point of which one is influenced by depolarization.
Okay, one more question.
Thank you very much for great presentation. And if the hex cell, when you express that, for example, the KIR 2.1 or some, such a potassium channel which keep the resting membrane potential deeper, the proliferation is reduced. My impression, I do such a thing all the time, and then my impression is that the expression of KIR 2.1 rather increase the cell proliferation. So there is some other way which may include the calcium influx, not through the voltage-dependent potassium calcium channel, but some other type of calcium influx.
I never tried that experiment, but I think this some other mechanism is happening in the other cell cycle phase. In my experiment, I only focus on mitotic phase because its cell is synchronized. So in other cell cycle phase, maybe some other molecule or some other membrane potential situation is important.
Great. Thank you very much to all the speakers. We close the session, and thank you.